Good morning, everybody. Thank you so much for joining us, for selecting us this morning. My name is Sira Echevarria. I'll be your chair today. And I will give you an overview of what's going to happen in this seminar. So, as you know, during this seminar, Spectronaut 18 will be launched. So we will walk you through the new tools, the new functionalities that it comes with. And I am very pleased to introduce you to the speakers. First, after my short introduction, Lucas Reiter, Chief Technology Officer at Biognosys, will actually be the one revealing the new functionalities, the new tools that come with Spectronaut. After that, we are very pleased to have two external invited speakers. Thank you, guys, for joining us. First, Greg Potts, senior scientist at AbbVie. He will show how they do their current workflow to screening small covalent binders to proteins, and how Spectronaut 18 is helping to overcome the bottleneck, the computational bottlenecks that come with big, large study sizes in drug discovery projects. And then Aline Martins brings a really cool project. She will present a project about how gravity affects brain organoids. And they are using proteomics, mass spec proteomics and DIA for this, and they also show Spectronaut for their data analysis. But first, I will give you a super brief introduction of mainly of what other software we also have to offer apart from Spectronaut. Everybody knows Spectronaut, I believe. At Biognosys, we offer not only software, we also offer peptide kits for different applications. And we offer software solutions for any mass spec-based proteomic application that you're doing. Our development, our products, our technology are developed based on three main pillars, which are performance, reliability, and ease of use. And we have, as I mentioned, software for any mass spec-based proteomics that you are running. Spectronaut is our software for DIA analysis. If you are doing targeted proteomics, we also have SpectroDive. SpectroDive has some functionalities that makes it better than some of the competition. And if you are doing TMT and you are still running DDA for either for TMT, for label-free quantification on DDA mode, Spectromine would be the solution to go for. Spectronaut offers the best performing library-free DIA workflow. For DIA, it's the known DirectDia Plus pipeline. It also comes with a versatile library generation workflow. It's the best performing solution for PTMs when you do that using DIA, and is continuously being improved and developed with artificial intelligence at the core of our roadmap. SpectroDive is our solution for targeted proteomics, and it is an unmatched solution when the throughput and study size is high. So if you have large panels of targets or you have a large sample cohort, SpectroDive is a very good solution to go for. And finally, we have Spectromine. So if TMT is your jam, or you are still doing LFQ in DDA mode, or you are running DDA to have spectral libraries and so on, Spectromine is the fastest and gets the best identification performance in the market. At least last time we checked. And if you want to get the experience yourself, we offer free of charge trials, always supported by our customer success team. We offer beneficial pricing for academic institutions, and if you get more than one, you get a very attractive discount. So if you're interested in trying them out, please reach out to us, talk to us here during ASMS, or visit our website, or reach out by email. And I wanted to highlight that we, the R&D department at Biognosys, they bring 3 oral presentations. So if you want to have a really high quality oral presentation at ASMS, I highly recommend these 3. Like this, you can see what we are doing on the R&D side, and what our technologies, how are developing, and how do we apply them. We also bring 9 posters These were too many to go through them one by one, but if you're interested, please ask or look for us in the program. You know how to do that. But yeah, very high quality posters. And with that, I want to give the microphone to my colleague. Thank you, Lucas.
Many thanks, Sira. Good morning, everyone, and welcome to our Spectronaut 18 launch seminar. I'm very happy to show you some of the new features of Spectronaut 18 and to be together here with two of our exciting speakers. So, in recent years, the throughput of label-free proteomics, DIA proteomics, has dramatically increased, as you can see in this figure here. This is on a log scale, and you can see that essentially from roughly 2012 to 2023, the throughput of these experiments has roughly doubled every 2 years. So this is quite an amazing achievement of the whole proteomics community, the mass spec vendors, the software developers, and so forth. And it also shows that the demand on data analysis has quite significantly increased over recent years. And so there is this trend about the throughput that I just mentioned. It's rapidly increasing, exponentially increasing. There is also a trend towards more complex data analysis. So there's a trend towards using library-free data analysis. We call it direct DIA. There's also a trend towards higher scan rates on the instrument, so more data is being generated, and as you probably all can see in your facilities, the files are getting bigger and handling the data is getting more complicated. So we have had a lot of customers asking for features how they can scale up their data analysis, and we have listened to them. So with Spectronaut 18, we are introducing Linux compatibility and also the ability to activate Spectronaut from the command line, which enables you to really program very complicated workflows in the cloud. So I can tell you a bit more about that. So you can imagine you have a facility with many instruments generating a lot of data, and you can push this data to the cloud, to kind of a central software that is going to distribute your jobs onto different instances in the cloud. And you can essentially, you can scale this up or down according to your needs. If you have very large experiments to run, you can scale it up. If you have medium experiments to run, then you can scale it down. Or if you have if your instruments are not running, you can just scale down your instances in the cloud. And Spectronaut already comes with quite a number of different tools you can use for that. So, or let's first start with what this master scheduler can potentially distribute. So you can potentially distribute full experiments in the cloud for analysis. That's the easiest because you have the least programming to do. But you can also distribute individual runs. If you have very large experiments and you want to scale up this processing, you can distribute individual runs, and you can do this either through direct DIA, so you can run direct DIA analysis on the different runs, or you can also run full library analysis on the different runs. And Spectronaut has tools that come with both of these ways to analyze the data. So if you run your experiment run individually with direct DIA, then you can just combine your archives to a library and make the full analysis afterwards, as you can see here. Or if you are doing library analysis, you can use all the SNE files and use a workflow in Spectronaut that is called SNE Combine to make the final analysis of your experiment. So Spectronaut is already coming with quite a lot of tools that support that. And of course, this part here is the parallelized part, so you can really scale it up to your needs. We are also working on something that we call Spectronaut Job Monitor. This will be launched very soon after ASMS. It's still in alpha mode at the moment and being tested. But what it essentially does is it will monitor a directory for incoming files from your instrument, and then it will analyze the files one after another as soon as they have been acquired using direct DIA. So your analysis is going in parallel to the acquisition, and then you can use the same trick that I've mentioned to you before, combining these archives and doing the final analysis. So it's a really handy way of doing your analysis while you are acquiring your data and saving time like that. This is how the UI is going to look like. So you have some features that you can use regular expressions and so on, that only the right files are going to be analyzed. And you can also see what you queued in the pipeline perspective of Spectronaut here. So switching to a different topic. So this is a topic that interests me a lot personally. So we are generating a lot of data, and if you look at this data, this is a plot I have taken from Grzegorz, who has a talk later at the conference about this. You can see that there are a lot of signals, these gray signals here, and we can only identify a very minor fraction of these signals. And I can also zoom in a little bit here on this particular data set, so you can see that in this specific DI data set, only roughly 3% of all the signals have been identified. And while this already leads to a tremendous amount of information you can use, it would still be nice if we could also go deeper into this gray part of this distribution here and analyze more peptides and extract more information from these experiments. And we are constantly working at that at Biognosis, as you can see in our latest releases. And again, in this year's release, in Spectronaut 18, we have been making heavy use of machine learning and transfer learning to improve what we can extract out of the data. And we actually also have a poster about this from Alex later in the conference. And this translates again into quite significant improvements in identifications. Here you can see depicted for the 3 different mass spec vendors, for Bruker, SciEx, and Thermo. So on protein level, we have between 5 and 10% improvements again for identifications. But of course, what most of you are interested in is not qualitative identification. So almost no one, or most of the projects are not that way. Most of the people want to work quantitatively and want to extract quantitative information from their projects. So, and we are quite picky about how we develop and how we evaluate whether quantification has improved. And this figure here is sort of trying to illustrate that. So what is better for you? Do you feel like it's better to have a very precise but not so accurate workflow, or do you feel it's better to have a very accurate workflow but not so precise workflow? And we are thinking a lot about how you can actually benchmark this the best, and I'm going to explain a bit how we are doing this in our lab to evaluate Spectronaut. So we are using a lot of these controlled quantitative experiments, how we call them. This is a concept that has been published many times. You're mixing different proteomes in different ratios, and then you have kind of a ground truth data set, and you know how the proteins are regulated in these data sets. Then we analyze the data with Spectronaut. These days, this is usually with direct dia. You get your quantities, your raw quantities out, you perform your statistics. Usually we perform very simple t-test statistics. And then what you can generate are these what we call candidate list curves. And what they essentially show is your candidates ranked by p-value. And based on the ground truth, you know how many of the candidates are actually truly differentially abundant proteins. And then you can perform some statistics on these candidate list curves. So we are using, for the accuracy metrics, we are using how many proteins have a fold change with an error smaller than x. This is the metrics we are using. For precision, we are using how many proteins have a CV smaller than y. And then we can use the combination of the two, which is our most powerful benchmark, which is how many truly differentially abundant proteins do we have with an error rate of Z%. And here you can see the results that we got from Spectronaut 18 compared to 17. We again have quite significant quantification improvements in terms of accuracy, in terms of precision, but most importantly also in terms of candidate recovery at 10% error rate. So we are really happy about that because it's quite a significant improvement. We also have been working quite a bit on the user interface. So here you can see a couple of examples. We have improved error messages, warnings, if you do very extensive searches, such as unspecific searches. We also have a special visualization how Spectronaut can identify peptides from chimeric spectra. So this is a new visualization that is very interesting. Then we also have a feature that if the license is expired, Spectronaut automatically turns into a viewer. So that's probably very handy for some of you. And then also all of the box plots, they have now Weiland plots as an option. So in summary, we really worked a lot on the scalability of Spectronaut because the trend is towards high throughput workflows quite dramatically in recent years. We again increased the proteome depth that we can achieve with DirectIA or DirectIA Plus. And more importantly, I would say we also worked a lot on the quantification, on the precision and accuracy we can get from these experiments. And again, what is very important for us is, of course, the user interface of Spectronaut, because a lot of people like the ease of use of Spectronaut. And yeah, with this, I want to thank the Biognosys R&D team that you can see here, and more specifically the bioinformatics and AI team that you can see highlighted here. Most of them are here at the conference, so talk to them if you're interested in what they are doing. And with this, I want to hand over to our two speakers, and thanks a lot for your attention. If you have questions, I'm not sure whether we take them at the end. Okay, thanks a lot.
So now we follow with Greg. Thank you.
Sure. Everybody can hear me okay? All right. So thank you to Biognosys for the opportunity to present today. Thank you all for coming this morning. So today I'll be talking about Spectronaut 18 and the recently released AWS version of Spectronaut 18, which we were able to benchmark prior to the conference, and the application of Spectronaut for activity-based protein profiling workflows, where we've basically translated those workflows into DIA-based analyses. So a question that I sometimes get is, why is your industry interested in AWS computing at all? And I think one of the assumptions that people make is it's pharma, so you have the money to buy the computers that you need. It's a very easy process. But computers are expensive, especially the computers that you need for computationally intense workflows. And so what we see, especially as lab sizes grow and there's more total users, I'm sure others in the crowd can sympathize that you have multiple users trying to use the same data analysis computers. There's not enough of those computers to go around. So there's conflicts in terms of scheduling. You can also have data storage issues where data files aren't being removed. So there's really a limited capacity for individuals to have their own high-powered PCs. So a solution that we and others are interested in is Amazon Web Services as a solution for this, because there is a lot of customizability with cloud computing. So for those who aren't familiar, a very big portion of Amazon's actual business is cloud computing and hosting of cloud computing. And so what I've shown here is for over 500 different EC2 instances of different types of computers, basically, that you can host through Amazon. You can see here along the y-axis, this is the total number of different instances that they offer, and they all charge at a different rate. This is in US dollars, so based on the power of the computer that you're hosting, you get charged hourly, so that can range from a couple of cents per hour all the way up to several $100s per hour based on the power of that computer. And you'll see here I've plotted also the virtual CPUs, so it scales with the number of CPUs that you're using. There's other types of factors that also can affect the cost. But for the demo that we performed, we used two different computers, one that was pretty mid-tier and then another that was a little bit better than mid-tier in terms of the power that it had. And so $3 to $10 per hour for usage. And so one of the nice things about the cloud computing too is you might be thinking, well, if you're hosting that computer that's hourly charging 24/7 for a full year, that's going to add up in terms of the cost. But you also are able to start and stop these computers remotely. And so you're only actively being charged for computers when you're using them. In this case, when we're actually performing a search. Then you can turn off the computer and you're not being charged anymore. So projecting the cost to actually host these computers and perform our searches through either Windows or Linux versions, you can see the cost varies. Windows is very popular, so the cost is higher. But over the course of a year, max, you'd probably be hitting about what the cost of a nice PC would be, unless if you're using Linux. So there's probably realistically an inflection point where if you're doing a lot of searching, it probably makes more sense to buy a PC, but it is an option at least. So in some of our benchmarking of Spectronaut 18, we were comparing to one of our lab computers here. So we had 8 cores, 96 gigabytes of RAM, and we were comparing to 2 AWS instances. Both had similar specs here, so you can see 96 CPUs and 192 gigabytes of RAM. And so comparing to our lab PC, we were able to compare 1 file, 5 files, 10 files, or 96 well plate worth of files. And so you can see at the low end, you're saving some time for sure, but about 10 files and above, that's where we see, compared to our in-lab PC, the number of the search time drops. And especially at 96 files, you're saving 1.5 to 2x in terms of the search time with Spectronaut 18. And so it's when we've made the comparisons to computers that are similarly spec'd to the AWS instances, we get identical results, identical search times, so it just provides you more options in terms of your data searching. So our vision for this, we have a lot of the pieces for this workflow built in place already. This is what we envision for automated data processing. We would collect with our instruments in the lab on the instrument PC. We would transfer to a data archive computer. That computer will be set up so that it's performing HTRMS conversion. So basically pre-processing some of these files that are then transferred to a storage server also on AWS. All of this would be collected every dozen files, or we could also do it time-based. But then, as was shared earlier, you could perform this in such a way that then you could be automatically searching data files that are dropped into a folder. So it really automates the search process. Then we can take those files out, load them onto a local PC for data viewing or automated data visualization that we can upload to a SQL database to share with lab scientists in our lab or others in the company, and then iterate on the process. So I think the most exciting aspect of this to us is that it could all be automated so that we're not having to transfer data and set up searches manually. We've also tested Spectronaut 18 compared to previous Spectronaut releases, and so as I'm sure everyone's aware, Spectronaut 16 to 17 was a very large jump with direct DIA. We still see increases Spectronaut 17 to 18 for thermo raw files collected with degrader proteomics. We've seen about a 2 to 6% increase in terms of the precursor IDs, and about a 2% increase for protein groups. In general, what we're seeing with direct DIA and how we typically take in DIA studies, with new studies where we don't have cell lines or tissues that we have very large libraries for, typically we'll perform direct DIA first as a way to validate new biology for collaborators. Then if we deem that we're seeing the changes that they expect, that's when we'll proceed to actually make a library. So it helps us make decisions in terms of how we're using our time. So now I'll pivot a little bit into how we could use some of this automated data searching. So we and others in the field have been interested in activity-based protein profiling developed from the Cravat lab. And so as an industry, we historically have not been thrilled with covalent small molecules for drugs, but there are a number that do exist on the market. And in recent years, the interest in covalent small molecules has increased again, especially because we now have these profiling techniques that allow us to look for covalent small molecules across the proteome. And so just to give you a brief overview of the traditional way of doing this versus the way that is increasingly being adopted, traditionally, when you're developing a high throughput screening campaign in the pharmaceutical industry, there would be a target that's selected based on prior biology in the literature or POC data that's been generated. Then we have biochemists that are making those proteins or expressing domains of those proteins. Then we'd run a really large compound screen of covalence or non-covalence. For a new target, a couple of months later, you'd go through exactly the same process. From target 2 to target X, target 100, it's very iterative. So obviously this is very redundant. It takes a lot of time for these screens. So the new kind of approach that many different companies are adopting is this in-cell screening approach, where basically instead of generating individual proteins, you're basically taking cell-permeable fragments, small molecules, and comparing to cell-permeable probes, where then you can query a single screen against the context of the whole proteome. So you're within the context of the cell, you have native structure there. Any hits that you have from that campaign then you know are selective for that protein target against the rest of the proteome. So you have that additional information available. And then many companies are in this space, startups, large pharmaceutical companies, so it's a very active field. We follow a very traditional workflow for this work, except we do this all by DIA. So to briefly give you an overview for those who aren't familiar, you could treat live cells either with DMSO or with fragments. So in the DMSO workflow, you treat with a cell-permeable probe. So this is an iodoacetamide disthiobiotin. So the iodoacetamide is going to react with cysteines. The disthiobiotin is an enrichable handle. So that probe is going to react with available cysteines. You then digest proteins into peptides. You do a streptavidin enrichment. So you basically have a cysteinome enrichment at this point that then you can analyze by DIA. In the fragment treatment, you have the fragment that you treat with first, followed by the probe chaser. So the fragment's going to block free cysteines. The probe is going to come in after and hit whatever cysteines are still present non-specifically. Digest into peptides, enrich, you'll see that there's a loss of this green peptide because our fragment bound. So then when you perform DIA and do the analysis like we do in Spectronaut, basically for a non-hit, you'll see probe-bound blue protein identically, so there's no change. When you have a hit, you have a loss of signal because the fragment basically competed with the probe that was present in the sample. So you calculate a competition ratio based off of this, and that's what's showing you the specificity for a hit. So the workflow, we made many different types of optimizations in our lab to try to expedite this process for DIA. I don't have the time to cover everything, but some of the biggest changes that we made are shown in the orange. And I'll discuss some of these in the next couple of slides. One big change that we made was in terms of the sample cleanup as part of our sample prep. There's a lot of different ways that you can clean up samples like methanol chloroform precipitation, but as you might imagine, this is a very manual process to isolate this protein pellet. We transitioned to sp3 beads. It's basically a magnetic bead that will bind proteins. You can wash away contaminants or reagents from your sample, and you basically do an on-bead digest from those beads. So you're able to enrich peptides that way. So comparing chloroform methanol precipitation to sp3, we have very good recovery and very good overlap, but the really important piece was because these are magnetic beads, they're able to be enriched, or they're able to go through automation. And so we've been using an Integra robot for this, shown at the right. You can see the number of wash steps that are associated with the sp3 cleanup, our streptavidin-based enrichment, very extensive wash steps that, if you're doing a 96 well plate, would total to over 1,600 wash steps. So the automation has really helped us with this, where we can go plate-based with these preps. So we have compared to TMT workflows, compared to our DIA methods. And I don't have the time to talk about all the comparisons that we've made, but I think just to say the TMT workflows do work. I mean, there's plenty of publications that show that. In our hands, especially for known targets that we have chemical matter for, you can run these studies both with fragments and with elaborated compounds. In this case, this was an elaborated compound. We knew what the target was. And then when we analyzed by TMT, the figure's a little compressed here, but you can, basically this was a compound progression where we would expect, compared to our control, we'd see greater competition over the course of the series. With TMT, we saw that there was just very wide error bars. Even when you were doing targeted mass spec, there was a lot of variability. With DIA, we were able to see a greater dynamic range. You can see the tightness of the error bars here. And we were able to remove some of the extra sample handling that's required for TMT labeling. So it's been beneficial for us from the dynamic range standpoint, and also removing the co-isolation issues that we all know are part of TMT analyses. So we're running all of our samples on the EvoSep, and so we've been able to get to shorter LCMS runtimes. If you compare it to one other landmark study in the field, we're able to get comparable numbers of sites with DIA. And so the workflows that we have, we collect our DIA raw files, we search with Spectronaut, where we've loaded the probe as a chemical mod within Spectronaut. We already have DIA libraries that we've generated for the cell lines that we're screening in. And so we would identify and quantify, we generate Spectronaut report files that then we can take into some of our own workflows for custom visualization. So Ryan McClure in our lab has designed a system where we can upload an experiment template file. We'll remove any site that has too many missing values. We'll impute whatever values we need to, and it'll do the normalization, all the scoring, and then upload to a SQL database that we or others in our company will use for visualizing the hits that we have from these screens. And so, just another numbers comparison, I guess, but we get comparable number of sites to very deep TMT data sets, both in terms of cysteine sites, but also proteins that are identified. And so finally, I'll just show one example where we were able to validate that the system actually can pull out meaningful targets. So Ryan in our lab was able to scrape UniProt to say for cysteines that we know that we were binding on proteins of interest like kinases, where are those cysteines located, just to help prioritize hits that we're able to identify. And so for MAP2K4, one example, we had a fragment that was hitting at 2 different sites on MAP2K4. One of those was in the active site of the kinase domain, and the other one was in just a separate part of the protein. So we had really good competition in the kinase domain and very poor competition elsewhere. So we looked at this fragment. We also found some other ones that had been hitting similarly. So fragment X we were just looking at hit MAP2K4. With these scatter plot visualizations, you can see cysteines that are basically being competed against the context of the rest of the proteome. So pretty good binding for MAP2K4. We also saw TNK1. We had fragment Y that was unique for MAP2K4 versus TNK1. And then we had fragment Z, which is just specific for TNK1. So these are some of the sites that we chose to use for proof of concept validation. So what we did was actually a titration experiment where, again, using our screening platform, we varied the dose at which we were performing our profiling. And so you can see for fragment X and fragment Y, we see really good scaling of the competition ratios with increasing doses. And fragment Z, which was not specific for MAP2K4, we don't see anything at all. We found a Luminex luminescence assay, or excuse me, a Promega luminescence assay, where basically we could take the exact same fragments and we were able to see similar competition at the high doses for the fragments. Compared to staurosporin, you can see a much stronger inhibition, but these are also just small covalent fragments, so we don't expect them to bind very strongly. And compared to fragment Z, we don't see any binding at all. So this was helpful to us to say, yes, we can screen, yes, we can detect biologically meaningful changes. So our throughput has increased specifically for DIA with all the changes that we've had over the course of our optimization. We're now able to run close to 500 samples, or this is now 800 samples within 30 days. And so throughput, we're expecting to only increase. We have timsTOF Pro 2s that we now have installed as well, which are enabling us to get to shorter runs. And so compared to Lumos or Explorer systems, we're able to see 3 to 4 data points per peak with our DI methods, even with wide windows. Going to the timsTOF Pro 2s and varying gradient length, we're able to get increasing data points per peak, up to 10 points per peak in some cases, which just helps with the quant overall. And then we're excited to see what it can do in terms of probe-based detection using CCS values with ion mobility. So I think in general, we're very excited about the new platforms that are being released as well with the speed that we're able to collect this data at. We're hoping to get to shorter runs and improve the quant that we're able to get out. So that's all I have for today, but if there's further discussion, I'll have a poster on Tuesday. I'm happy to talk more. I think in the interest of time, you already heard all of this, so these are just my conclusions of what we discussed today, but I'd like to thank my colleagues who were integral to the optimization, but especially Ryan McClure and Alex Shannon on this list who were integral to the work. And with that, that's all for me. I think we have questions at the end. So, okay.
Thank you so much, Greg. Nice presentation. Now we move forward with Aline.
Okay, good morning. Thank you very much to be here so early. I know that is kind of rough on such an early birds here today, but I really appreciate that because today we will talk a little bit more about brain organoids, so you will understand how this microgravity can affect these organoids. And I would like also to thank Biognosis for the invitation. So I'm very glad to be here. So today we will talk about an experiment that we shuttled to the International Space Station to understand the role of microgravity in neurodevelopment experiments using brain organoids. And for this experiment, we use a lab that they call Cube Lab that stays inside the International Space Station. So I will talk about this today. So how this project began? So this project began because we have a strong collaboration with Dr. Alison Watry from Watry Lab. He is a scientist that developed the experimental model of cortical brain organoids. And he was already working with Space Tango. Space Tango is a company that supplies ISS, giving engineer and biological solutions for ISS. And they were already working on RNA sequencing. And then Watry, that has a long collaboration with Yates Lab, asked, how about the proteomics? So we understand a little bit how microgravity affects these models, but we don't understand how it affects the effectors, how the effectors are changed by microgravity. So this begins our work with Watry Lab and Space Tango, and then finally we were able to shut the experiment on this mission from SpaceX. CRX-25 was an ISS resupply mission, and then we used the Cube Lab. This is the Cube Lab that stays inside the International Space Station. Sorry. So this lab was really interested for us because we would like to just isolate how the microgravity events were affecting the brain organoids. And this Cube Lab permits this because it's made by lead and isolate the radiation outside the experiment. So it's super heavy and it's difficult to manage to put outside there because it's kind of a heavy load. But we managed to talk and to have this experiment on ISS. So this was the beginning of the story. So what we did was to use brain organoids that were generated by iPSC fibroblasts from patients that had Rett syndrome. Rett syndrome is one of the diseases on the autism spectrum syndrome. So we generate neurostem cells from these patients to build the brain organoids. And we did this in triplicates, and also we had access to control patients. These control patients are patients that are directly related to the patients with Rett. And then we did the same thing to extract the fibroblasts and to generate the stem cells. So this is really important because these controls are isogenic. So we didn't have any kind of bias on this. So of course that we set one experiment to go to International Space Station in the same time that we had a ground experiment here, because otherwise we wouldn't understand how microgravity would affect this model. So then when this experiment stayed 30 days on the International Space Station, and then after this 30 days, nearby the mission come to Earth, we fixate these organoids up there, because there is some effects as descendant effects that could affect the brain organoids. So we decided to fix before the descendants. So these brain organoids were harvested from the Cube Lab, right in Earth, and then we were running the same experiment on ground experiment on this, and then we applied the sample preparation for proteomics. We use EvoSAP as a chromatographic method. We use TIMS-Pro 2 as an analytical method, and we acquire the data by DIA-PASEF, which is a data-independent acquisition. So, of course, we use Spectronaut to analyze this data because, as you can see, the samples are really rare. And it's really difficult to place an experiment there because you have to use the cube lab, otherwise you won't have the isolation from the radiation. So we needed to use a software that was robust, and we could retrieve the information back after this analysis. And we also were interested to generate some functional insights about this organoid over there. So we tried different methods to acquire the data, but actually the best method that worked for us was the 30 sample per day in a 15 centimeter column on the EvoSAP using Spectronaut as the software to analysis. So we had to have some strategies because as you can see, 30 days, it might be not enough to evaluate how microgravity affects these organoids. So we were really afraid that the difference was so subtle. So we decided to also have different strategies to increase the identification of the proteins. One of them was, of course, we analyzed the brain organoids one by one, but we also had the pool to increase the signal of this differentiated proteins. And that was really interesting, is a strategy that we are using a lot in single-cell proteomics, is to build a tailor-made library that is from the same cell type of the brain organoids. And that was a very fortunate strategy that we use. And again, it was possible to integrate this hybrid library through Spectronaut. So this really helped us to identify these proteins that were analyzed on the brain organoids, using also the direct DIA. So the first thing that we would like to understand is how different these organoids that were in space are from the ground experiments that we have in Earth. Because we have been working with brain organoids in Rett syndrome with Muoto for a long time, Yates lab. So, but we didn't know how this organoid behavior over there. So that was the first thing. And one thing that struck us was that we had a lot of differential proteins on the organoids compared to the ground experiments. So this first evaluation was just brain organoids that were in space, but they were very different from the ground experiments that we have. And we have this variety, and the intensity were much more intense, these proteins, under the microgravity events. So one of the things that one of the cluster of the proteins that were enriched was proteins related to transposon elements. And transposon elements are really important in neurodevelopmental diseases. And this was really intense on these organoids that were suffering microgravity events. Also, proteins related to cocaine syndrome, which is also a neurodevelopmental syndrome. So that was not unexpected, but it was very intense, this proteins there. And also proteins related to the synapses organization. So we did also an interactome, a map, and again, on the biological function, we see a lot of proteins related to replication and metabolism of RNA, peptide elongation, and also some proteins that are related to deacetylate stones that are responsible and they were impaired on this experiment that they are related to neuroplasticity and memory in brain. So, of course, that we compare the ground experiment to the space experiment, especially our interest is what to understand how these two organoids behavior within the Rett syndrome, the organoids that were carrier of this deletion. So again, when we compare the space organoids with the ground organoids, we see an intense replication. We see a huge variety of proteins that are present on these space organoids and are not present on the ground experiment. When we generate the heat map, we even see proteins that were kind of absent on or in very low quantity on the ground experiment. And so the same, I mean, proteins that were absent in this space comparing to the ground experiment. Remember that we are comparing brain organoids that had the same syndrome, Rett syndrome. So again, on this experiment comparing microgravity and ground, we see a lot of proteins related to the metabolism of RNA, transposons again, but not being the first enriched biological function. And we also seeing vesicle-mediated transport proteins related to this gene ontology and exon guidance. That was interesting because, of course, there is a lot of, not a lot, but there is out there already some experiments with astronauts and that suffer from microgravity. There is this interesting experiment with twins, and one of the astronauts were in the International Space Station for 1 year, and the other one was in the ground. And then we see the molecular functions that are impaired on these brain organoids that leads to the same phenotypes that were impaired in this astronaut that were there suffering from microgravity events. Again, when we use the interactome, we can see that this mainly for biological functions were enriched in this comparison. So just to talk a little bit about this main impaired pathways. So transposons are really well known in Rett syndrome, but they seem to be more, there is a higher expression on this kind of transposons on space organoids. Some of the proteins were related to DNA repair. They were impaired also, and this is also seen in this syndrome called cocaine syndrome, which is a neurodevelopmental syndrome also. So effects on ribosome activity and peptide synthesis, it seems that they are increased in microgravity. And again, I have to tell you that these are preliminary data. We didn't have a lot of time to deal with the data, but they are really related to the things that we are seeing, the phenotypes of the astronauts. Effects on synapses organization, microgravity lead to alterations in neuronal morphology, especially important structure for synaptic connections. So we are trying to go deeper on this feature. Effects on trafficking, vesicle-mediated transport. It seems that microgravity might be involved with the accumulation of neuronal-derived waste. Impairing transcellular degradation that leads to increase in vesicle-mediated transportation. And effects on axon guidance. It seems that microgravity, again, can interfere in the perception and interpretation of important microenvironment cues around on this organoid, which is a 3D culture. And also, Water was interested to do RNA-seq with the same samples that we have. And then, again, we have the same impaired pathways. So that was really interesting because there is some correlation. We are getting further on this data, but there is some correlation with the findings on RNA-seq in our findings. And in 2012, Gog related some dysfunction, especially with he also had some work with microgravity, and he found out that some of the proteins up in the space were truncated. So we are trying to see this also, if this is happening with our proteins, because we see a lot of replication on this comparing these two models. So, and actually we call from space, but actually the right term is LEO, which is low Earth orbit. It's not out of that in the space, but this, it suffers from microgravity anyway. So the conclusions is, in this preliminary work, microgravity might be responsible to identify the ribosomes, intensify the ribosomes and the RNA metabolism through a myriad of processes, impacting in the efficiency and accuracy of the protein synthesis. So we think that microgravity, even though in a very short time, 30 days, but is still an important event on these models of brain organoids. The studies conducted in the space have shown that ribosomes produce proteins with different structures and functions compared to ground experiment. That is for sure. This altered protein synthesis can result in change to cellular process and functions, especially in neurodevelopment. So, to conclude, I think that this triad, EVOSEP as a chromatographic method, Bruker timsTOF Pro2 as an analytical method, and the Spectronaut for data analysis was really make this, this work possible. But, as I said, this is preliminary data. Of course, we need to see the other stuff that is really interesting, let's say the PTMs, but we didn't have time to go further on this. This is a parallel project that I have. I actually work with single cells, but this is such a cool project that I couldn't leave behind. So this is a beautiful project, this is a beautiful ongoing project. And I would like to acknowledge, of course, the Yates lab, because it makes this possible, but this beautiful collaboration that we have with Muotri lab. And I would like also to acknowledge Bruker that helped me a lot on the analysis. And in the first step in Spectronaut, that was the first time that I saw the software and really, really helped a lot. So I would like to acknowledge Diego Assis and Aline Leite, which are two wonderful Brazilians at Bruker. So thank you very much.
So I want to thank everybody for being still here and for your very interesting questions, and very especially to the speakers. Those were really, really nice talks. We appreciate it, and please join me in thanking the speakers again.